Hi everybody, let me introduce myself. I am Nermin Əlesgərova. I am the teaching assistant at the Department of Epizotology, Microbiology and Parasitology at the Faculty of Veterinary Medicine. Today I am going to present my further lecture to our students and all of our listeners. The topic of our today's lecture is influenza and smallpox diseases in farm animals and birds. Throughout my lecture, I will try to give enough information about both of these infectious diseases. And throughout our lecture, I will give enough information about these important points. The first one is the etiology. But what is the etiology? When we say etiology, we mean the etiological agent or just agent or certain infectious diseases. Because the etiologic point or the etiological agent just own the certain infectious diseases. And second one is the epidemiology and the transmission. Epidemiology and transmission of certain infectious diseases is very important point because spreading infectious diseases around the world cause pandemics in many countries and around the world. And next important point, clinical findings or clinical science or the symptoms. During infectious diseases, it is easy to observe certain clinical findings, clinical science or symptoms. And further one is the diagnosis. For the diagnosis certain infectious diseases, we need some samples, some appropriate samples. By the help of these kind of samples, we try to diagnose certain infectious diseases. And differential diagnosis. Differential diagnosis issue is that we try to differentiate from these infectious diseases to another infectious diseases because sometimes the clinical findings or the clinical symptoms are the same in many infectious diseases. Sometimes it's not easy to distinguish them. And last one is the prevention and treatment. But what is the prevention and the treatment? Prevention or the prevention measures or the protective measures against certain infectious diseases. When we say prevention, we mean some immunizations, vaccinations, some disinfections, this kind of things to avoid certain infectious diseases. And last one is the treatment, to treat certain infectious diseases by the help of some drugs, medicine, some antibiotics, some bacteriophages, some interferon and anaferon, this kind of things. And this is the structure of the influenza virus. As I mentioned before, the topic of our today's lecture is the influenza and smallpox diseases. This is the structure of the influenza virus. And I can say that influenza virus is a type of the virus. So virus is the submicroscopic organism. That's why it's impossible to observe the viruses by the naked eye. For the observing viruses, we need electron microscope. And virus is the obligate parasite. That's why virus is different in comparison with another type of the microorganisms. For instance, and unlike the another type of the microorganisms, virus need some living cells organism to grow or the replication process. That's why virus needs some living cells or the organisms. When I say living cells of the organisms, I mean some kind of things. The first one is the lab animals. You know, there are lots of lab animals, but what are the lab animals? Lab animals are the animals, they are very sensitive to the influence of the certain infectious diseases. In addition to it, it is easy to obtain lab animals and it is cheap to obtain them. They are not expensive animals. That's why we use lab animals in the virology sections of the lab and another sections of the veterinary labs. And lab animals including mice, hamster, sometimes small ruminants, frog, sometimes dogs, cats, and this kind of animals. Everything depends on the sensitivity of the animals to the influence of the certain infectious diseases. And in addition to it, sometimes there are some type of the virus, they need cell culture. But what is the cell culture? Cell culture is a type of the culture. We located them inside the matrix, and we need sometimes the influence of the cell culture for the growing process of the virus, and the growing process of the virus is different in comparison with other type of the microorganisms. So the growing process of the virus is called replication, and inside the body of the lab animals, inside the cell culture, inside the embryonated egg or the fertile egg, it's possible to grow viruses, unlike another type of the microorganism. And after 1930s, scientists started to claim that using cell culture in virology section is the best way to grow viruses and for the isolation of the viruses. And the isolation issue of the virus is very important, because sometimes we need some type of the virus isolation, for instance, obtaining certain vaccine against some infectious diseases. And for another purposes, we need this kind of living cells of the organisms. And last one is the embryonated egg or the fertile egg. We need embryonated egg because of the growing or the replicate viruses, and sometimes isolate them. And in addition to it, sometimes we need fertile egg for the another purposes in lab